Thanks, Linda. And thank you, Professor Lorraine Martin, as well. That was brilliant. So my name's Jim. Myself and Gamma, who sat here at the front, are your first co-chairs for this session on innovation, entrepreneurship, and industry pathways for AMR. We've also got two moderators who, during the Q&A, will be handing around mics for asking questions. So that's Ahmed and Becca. So once again, as Linda said, if you can please just hold on to your questions or write them down in your conference notebook. We'll have a panel Q&A at the end. So please can I introduce our first keynote speaker, Dr. Hannah Leese from the University of Bath, who's going to be talking to us about from sensing to healing, an integrated approach to infection detection and treatment. Thanks, Hannah.
Thank you very much. Thank you. Right, so let me just check this days. Okay, cool. I can get that. So it says next and back. Okay, that's really clear. That's good. Wonderful. Thank you so much for that kind introduction and this really kind invite and opportunity and privilege to talk to you today. So the title of my talk is From Sensing to Healing, an integrated approach to infection detection and treatment. And what I'm going to talk to you about is how over the last few years we've approached this in combination to antimicrobial resistance and sepsis, and the motivations that I have in the Materials for Health group that I lead, and how we innovate different materials and devices to come up with solutions or potential solutions or platform solutions, which I'd love to hear from you, and ideas more so that we can really get them fit for purpose for fighting AMR. So, yep, I'm from the University of Bath. There's also a Centre for Bioengineering and Biomedical Technologies at Bath, and I'm part of that leadership team there. I just wanted to share very, very briefly just a little bit of kind of, because it's so multidisciplinary in this room, I just wanted to share a little bit where we are. So the vision of the Materials for Health Lab is to engineer advanced materials and devices that enable new forms of diagnosis and treatment, from human to environmental health. So in general, of course, from the agriculture and environmental AMR, also to human health as well. But also looking at that sustainability through a sustainability lens. So what materials are we using? What processes are we actually using to make these devices? What's the lifetime of them? Are we actually then going to make some other damage later down the line? So this is also something that we are working in the group too. So we are at the interface of quite a few different areas. I'm going to talk to you quite a bit about the healthcare device manufacturing today, and a bit about biomimetic functional materials as well. I'll touch a little bit about the sustainable polymers as well, and what I mean by sustainable polymers, but I'll mostly be focusing on these healthcare device manufacturing. All right, so again, I don't need to introduce this bit, I think, but I did want to connect the sepsis part to antimicrobial resistance. Of course, we are in World AMR Awareness Week. There's no coincidence that we've got this event in this week as well. But we're very also aware of this public health message, which has changed over the years, but is in the sense of it's visually changed, but it's still there. Keep antibiotics working, and how we can get that public health message across. But related to that, and very much related to that, is sepsis. And everyone may or may not be aware of what sepsis is, but it's when it's like a runaway inflammatory response by the body. So the body starts to, some people see it as like the infection of blood, but actually it's the runaway inflammatory response of the body. So then it starts to attack its own organs, and it is in response to an infection. It doesn't necessarily have to be a bacterial infection, but it is in response to infection. And it can be a very complex problem to treat, and generally there are a panel of antimicrobials that would be supplied, and the symptoms of sepsis are really difficult to detect. There isn't a specific biomarker, something that we could actually say, yes, that's definitely sepsis, and it can start relatively like flu or symptoms like that, and then it can run away very quickly due to the cytokine storm. So their evolution could be the end of ours, in the sense of when I spoke to a clinician who's a neonatologist, and we discussed about the connection between sepsis and antimicrobial resistance. And in the last 5-10 years, we had a conversation actually last week, and we met for the first time 5 years ago. And I asked him, have you seen in neonatal care in your neonatal care more resistance already in the last 5 years? And he said, yes, Hannah, I'm seeing it more, where it's getting harder we are seeing more resistance generally. But 5 years ago, we were talking about the urgent need to be able to detect this early and rapidly so that we can keep antibiotics working. So from how can I contribute, and how did I feel that I could contribute to this area? And this was around how we can engineer and innovate solutions to detect infection early and treat precisely, and have precision treatments in relation to early detection. Now, need innovative solutions that detect early and rapidly, accurately to inform treatments, simple to use and interpret, and truly independent of resource settings. Antimicrobial resistance is everyone globally, and everything globally can be affected. The acronym kind of fell out of what I'd written, which was a happy coincidence, but in the sense of it easy, not so easy. It's really not easy. It needs to be easy to detect and treat, but it's not. So we've been really focusing there over the last few years since I established the Materials for Health Lab. So just to kind of relate this to how I see it. So there is a cycle between why I've kind of talked about sepsis and antimicrobial resistance, because from sepsis is a runaway inflammatory response. It will need a panel of drugs and antimicrobials, which means that all of those drugs will be delivered, but not all of them will necessarily be useful. But all of that will then be exposed, and the bacteria may well develop even more resistance. Without having sepsis, we have prolonged exposure to treatment as well, because you need to consistently treat. And this lack of early detection of sepsis means that we can have delayed infection to detection, and then again get to a series of trying lots of different antimicrobials and treatment. So sepsis is a very complex inflammatory response. As I said, there's no currently single biomarker for diagnosis, and one of the ways that we look for whether sepsis, whether a patient is experiencing sepsis, and that's across ages, is to look at the different inflammation responses, as well as then taking blood samples and obviously culturing the blood to see whether the bacteria and other microbials grow. And that can take several days, and that can be too late, far too late. It needs to be really as early as possible, as early as it's presentable, and really thinking, could it be sepsis, and let's see whether it is. So in this topic, it's about innovation, and innovative interventions are very much required, but in collaboration, and definitely, I'm not saying I have the answers, I just want to contribute all the answers, but I want to contribute to make this impact. So one way that we've done that is really look at minimally invasive technologies. Now, in the previous slide, I spoke about it needing to be independent of resource setting, or at least able to be used in lots of different resource settings. So we really tried to simplify down as much as possible the devices and the technologies that we've been looking at. We've been looking about how we could think about manufacturing them in slightly different ways, in a decentralized way, or mass manufacturing, and then having it really cheap to be able to be deployable. Also trying to minimize electronics, trying to minimize all of those different aspects as well. So even though there is the smart part that could happen to these devices, I'm going to show you very much about the ones that we've really tried to make as simple as possible. So we've used additive manufacturing, 3D printing technologies, and also something called electrospinning, which I will introduce briefly at the end, but this is related to wound dressings, and again, having wearable, non-invasive devices that could detect and treat infection. So I'm going to talk to you about most of the talk is going to talk about one minimally invasive type of device, which you might have heard of as microneedle technologies. And Ryan Donnelly as well, who's based in Belfast, is very, you may know him or not, but worked a lot in drug delivery of microneedles as well. But we've looked at microneedles both in this drug delivery and detection, and why we would want to use those microneedles is to try to access the interstitial fluid. So one of the aspects of not necessarily having to access the blood for risk of further infection introduction, but also having more patient compliance, as well as being able to detect people with very little fluid to give, so very vulnerable babies, for example, and also not introducing pain, which we don't really understand the levels of pain of babies, that really what they experience. So we thought, how can we do this if there's no specific biomarker? So we've been thinking about looking at what we call as multiplexing, so looking at multiple biomarkers, because the levels of the inflammation-related biomarkers in the body, as a panel of markers, they change over time when somebody is in sepsis. So it wouldn't be one, it would be a panel that we would want to, what we would want to assess. So that's that detect and tell part. And then it's this deliver therapeutics as well through the interstitial fluid or through the skin, rather than through the mouth or intravenously. But again, the question is that systemic? Would it go everywhere that it needs to? And this is something that we still are working on. So just to go into a little bit more detail about what these minimally invasive devices are. So microneedles are sub 1 millimeter needles. You may have seen, actually, recently I went on Amazon and I put microneedles in and I saw some very scary looking microneedles. So it's not those that we're going for. They are really very tiny and they are very low force. So you do not, and you should not feel that pain from them. If you push really hard, it's possible. And they penetrate the first few layers of skin. And that gives us scope to detect and deliver on device. So what do I mean by that? So this is an example of a microneedle which is better drawing than me, so thank you, BioRender, for this. But we can look for different biomarkers, different panel biomarkers that can go up into these microneedles, and we can have sensing elements within the microneedle connected to the microneedle for detection. They work on very low volumes as well, and we have some signal. It could be colorimetric, so it could be a qualitative. I'm going to show you a qualitative signal, but we've also made it quantitative as well. So there's ultimately being quantitative for sepsis detection, for example, would be very valuable. But then in response to that as well, in that response to that detection, it's possible to use the same technology, maybe not exactly the same material formulation, but the same technologies to be able to drug deliver as well. So we've looked at I'm going to show you an example of a hydrogel microneedle, which is able to swell and release over time into the skin. And at the end of the talk, I'm going to very briefly show you about some of the work, which I'll be super happy to talk to everybody about later as well, but just in terms of time, about wound dressings and how we can also look at those as minimally invasive devices as well. Again, both for sensing and for drug delivery. So the advantages of microneedles I've touched on a little bit already, but this point of the small volumes that are required, improving patients' compliance. Needle phobia is a real thing, and it's something that many people will not have or go to or be very vulnerable or reluctant to take it if they see a big needle coming towards them. And pain-free, I put in kind of air quotes because, as I said, there are some microneedles out there that may hurt and may get the pain receptors, but the ones that we're designing are really meant to be low force, as I said, and not reaching those pain receptors, causing pain. Some of the limitations and it's a very fair question that I receive as well, is why are microneedles not yet in clinic? And actually, it's in the last 2 months, there's been a first FDA-approved microneedle device, finally, after many years. And there's different reasons for it, but some of the limitations have been expensive, specialized manufacturing, the microneedle mechanical properties, the way that, how do we extract it? Do we extract enough? The detection might be offline rather than exactly where you need it, so how much is it actually speeded up? and making sure that it's reproducible, sterilized, all of those problems that, that we've been trying to face as well. But the real key point from this slide is really to think how we can utilize minimally invasive devices for uptake, detect, and delivery. And we could also then use them to know when to stop also delivering. And I think that is something that is a utopian idea in my mind at the minute, but I hope we can get there. So in the sense that we need to know when the infection is clear so we can stop. And that's where that precision medicine comes in. So I'm going to very briefly say how we make these kind of very simple, straightforward, deployable microneedles. We use a lab bench 3D printer. This one is now relatively very affordable. We can make hundreds in one print for the same amount of time. And we played around with trying to increase the resolution of the printer, because one of the key points is that can it really penetrate skin, and are you able to get a sharp enough point to be able to hit the microneedle? So key design parameters that we explored was the polymer type, the printing angle, and layer height. Now you might remember at the beginning of the talk I said about this sustainability point. So right now, some of the ones that I'm going to show you are not sustainable polymers. So they are still non-renewable polymers. And from that sustainable lens, it's more about the accessible sustainability of healthcare rather than material design. So we take some CAD, these are computer-aided designs, CAD designs, and we were able to manipulate, they look pretty nasty from here, but we were able to manipulate the tip related to how we printed it and what angle we printed it at. And in general, then we were able to have a look at and manipulate different structures and really get beyond the resolution of the printer, of the manufacturer. But what we also use now in the group is something called 2-photon polymerization, so the SLA printer that I'm showing you with the roughness here of the lab bench stereolithography 3D printer is also a UV curing printer, but the 2-photon polymerization enables us to get even higher resolution. So very, very, very sharp tips. You can go down to the hundreds of nanometers, and this is just one example of the microneedles that we're working on. So we've also made these, you can see, are hollow microneedles, so they're like tiny mini hypodermic needles. And these ones are the solid needles, which we use as templates for other microneedle formulations. So I'm going to show you the first case study of detecting, and we used hollow microneedles here. So directly printed to integrate lateral flow devices. So this was, I think we all got very familiar with lateral flow devices during COVID, but there are lateral flow devices for procalcitonin, C-reactive protein, and other inflammatory markers as well. So what we wanted to do was make it as simple as possible to be able to take up the interstitial fluid through the microneedle onto the lateral flow device and see whether we can detect these inflammation markers through this device. We had to do a functionalization, so we had to change the surface chemistry to make it hydrophilic enough. There is no there's no pumps here, there's nothing, it's all capillary action. So we had to change it, the surface chemistry, to make it hydrophilic enough to be able to uptake spontaneously to then take onto the lateral flow test. So in general, I don't know if I can do this, maybe that would be better. Yeah, in general, so this hollow microneedle here, we coat it with a peg on all sides, and then you can see here, this is the 3D printed without coating. This is plasma treating, so this is making it hydrophilic, but only for a short amount of time, but you can see here. Then the pegylated, so this polyethylene glycol hydrophilic polymer then maintains for 30, well, this one was 30 days, but actually 90 days and over. And it also is possible to go through autoclaving, sterilization as well. And then we also test the mechanical testing as well as integrated this lateral flow test at the limit of detection of the inflammation-related biomarkers for those lateral flow tests. Okay. So what I want to show you here is just that, yeah, it does, because one of the biggest things for us was that are these microneedles able to actually pierce them, or do they just dimple the skin? This was a real important point for us, especially from printing directly from the printer. So what we see here is the displacement of the hollow microneedles so that they're not collapsing. They have some movement. They displace by about 25 microns, but they're in total about 700, 800 microns in length. So very little movement, they don't break at this point. And we focused on this force per needle to try to get as low force for penetration as possible. So we want to make sure that there is minimally, we're pressing as little as possible, because our thumb force is actually much, much larger than we would need for the penetration of these microneedles. And what we're showing here is that this coating that I showed you for making them hydrophilic and making sure that we have this spontaneous uptake of interstitial fluid, it also does not change the displacement of the needles. And this is just an example of, this is ex vivo porcine skin, and the array holes from the penetration, and the histology to see that it is actually pierced rather than just dimpled. So just to kind of show you how it works. So we've got our C-reactive protein, which for the lateral flow test is 10 milligrams per mil, the limit of detection of the lateral flow test, and procalcitonin is 1 nanogram per mil. And we simulated the ex vivo skin as a, so the molecules and the fluid have to go through the ex vivo skin to be able to be detected onto the lateral flow test. Now, I don't know whether this is going to work very well because I haven't got access to and I've got bookmarks through the video, so I will show you later if you want to see it later, but as you can see, I don't know whether it will go to each one. But what this video shows is that we're looking at CRP. This is a control here. And then as we go through the video, what we see is the hollow microneedles. This is an untreated hollow microneedle here. So we don't see any positive result. And then when we see the treated hollow microneedles, we then see this positive result onto the CRP and PCT. But what you can see in this image is that they're mixed. So what we did as well is we combined the two. We had 2 lateral flow tests and multiplexed this. So we were able to detect both molecules at the same time at their limits of detection of the lateral flow test. Again, proving that concept that it is possible to have really a straightforward, printable, easy device and be able to start to detect these inflammation markers relatively quickly. Oh, thank you for moving it along. I really appreciate that. So this is the pegylated coated ones, and Joe, you can see here that they're positive. And yeah, and then this one is the double one. Thank you so much. This one was a bit more fiddly to put on, but in real life, if it was actually in clinic, we wouldn't have it so large, but it was just for ease of the lateral flow test dimension. And then if it can run to the end, if that's possible. Fantastic, thank you so much. And again, both of them are positive here. So if the patient was in hospital or at the pharmacy or so on, this will be done in less than 5 minutes. This was up to 5 minutes per run, so it is relatively rapid and quick. Another one as well is about conductive microneedles. So what we've tried to do is make them, I said that was very qualitative, and this is quantitative. So we're trying to move towards quantitative detection. In this case, then, we looked at taking away the metal as well completely. So again, thinking about resource setting and putting completely conductive polymers on here. And I've got my time here, and I know I've been rabbiting on, so I'm going to move on to this part. So what we all 2 minutes. So what we also do is modify the microneedle with something called a molecularly imprinted polymer. So I said I would touch on a little bit about the biomimetic polymers. So it's a synthetic antibody. So again, from a resource setting stability, if it's cold, if it's wet, if it's really hot, it should be stable. It doesn't need any special storage. So in general, this is one of the advantages of molecularly imprinted polymers. So what we can do is imprint different molecules, and in this case, cortisol is one example, but what we have also done is imprint very specifically E. coli and Staph aureus in these different sensors. Not directly yet on a microneedle, and you say, Hannah, we can't detect bacteria necessarily with a microneedle. Indeed, but from taking a drop of blood and making this possible, we've made it really specific for gram-positive and gram-negative bacteria. And this is an example of how sensitive it is. So an aptamer, we bind to the LPS of the lipid polysaccharide of each of the bacteria, and it's very, very specific. Combine that with a polymer, then we start to get some really large differences between our bacteria and the controls. So we are able to very easily distinguish between gram-negative and gram-positive bacteria. I'm very happy to go into more detail about that, but due to time, I won't say that. And the last part is about the treatment. So here we use this solid microneedle technology of printing, so easy printing, and then do a molding. And this is an expensive stereolithography, a photolithography template, and we were able to 3D print and replicate. And in this case, we took up amoxicillin and vancomycin in this case, the two very commonly utilized antimicrobials, and were able to also penetrate the skin in this case, and the amoxicillin stays active. I know these are just simple agar plates here, but this is where the hydrogel microneedles were placed, and again, we're using this with E. coli and Staph aureus, and the hydrogel drug is releasing and killing this bacteria over time. So coupling these two technologies, or the same kind of platform technology into different application for detect, treat, and tell. And my final point is really quick, so I'll just go through these, is about the nanofiber wound dressing. So again, another type of area of work that we're doing. We can make these nanofibrous mats detective as sensors as well. So for example, the aptamer molecularly imprinted polymer I just showed you, we could coat our coating onto this nanofiber as well as this treatment. So here we have tetracycline in this case embedded into a sustainable polymer. So this is a completely renewable polymer and biodegradable, and could be used as an effective wound dressing for treatment. And the reason why I call it Janus is because of the different wettability properties of these two. So we are able, one is hydrophobic and one is hydrophilic, so one is a barrier to the wound and one is the other layer is treating the wound. And there is effective killing, and the tetracycline stays active after this electrospinning process. So these could be bandages or wounds or wearables if required. So with that, I'm looking towards 2050. We need early, precise, and rapid detection of infection at point of care, and it's crucial to relieve the burden of sepsis and fight antimicrobial resistance. And by designing minimally invasive technologies, we will hopefully enable that early and rapid detection and make that applicable in all settings. Looking at that material design, this antibody biomimetic, looking at how we can replicate that to make as stable devices as possible. And as a final point, to innovate sustainable material synthesis processes, so really look through the whole process and life cycle of these devices to ensure that the sustainability of the material synthesis upstream, but then also what happens to that material once it's been used and downstream. And with that, thank you for letting me go a couple of minutes over. And I will say a huge thank you to all the Materials for Health Lab and collaborators. And yeah, thank you very, very much for listening. Thank you.
Thank you, Hannah, for that really interesting talk about that novel technology. So